Okay, so this is the first lecture for the class. We're going to talk about chemical basis of biomolecules as well as bonding in the biomolecules. I have uploaded the PDF file for this lecture into the files section of the Bio courses. I also have broken down the lecture into two pieces, so we're going to talk about enzymatic interactions in a separate video, which also will be in the Bio courses. Before we go on with the lecture, I wanted to say that I really like this book, but this is not an introductory book per se. It doesn't dump things down like a lot of other books did, and as a result, we're not going to be covering everything necessarily to the depth that the book covers. But everything that appears in the lectures will be in the exams. I'm not going to try to make your lives hard here, but it will be sufficiently challenging. So also, there's homeworks at the end of each chapter, and there's answers at the end of the book. So I recommend you doing practice homeworks to prepare yourself for the problem sets that will be due before each exam. The quizzes will be based very much on the lectures themselves, as opposed to on the book material. Also, I will upload this lecture as well as all the other subsequent lectures into the files section of the Bio courses. And if you guys have questions that you can't ask in class, obviously, because this is just a video, you will email me directly to my UIO account, and I will answer the question and probably also post the question and the answer in the discussion area at the Bio courses so that everybody can look at it and read. Now let's get going. So biomolecules, I'm sure you have covered in Life 1010 or Biology AP or whatever other classes you have taken. But I wanted to take a little bit of a different approach. We usually think of molecular biology as so completely removed from real life. But in reality, just the simple food pyramid that you see here covers all the biomolecules that are essential to us. Food is essential. We need food to survive. And we need biomolecules. We need to take in different biomolecules in order to maintain all of our cells. That's the reason we eat. Unlike plants, we can't generate our own carbohydrates out of from light and oxygen, from light and CO2. So let's look at the biomolecules as represented in a food pyramid. So there's the lipids. That's the fat and the oils. They usually, when we take them into the diet, they're fully refined. They only come as triglycerides. There's nothing else attached to them. The fat cells that were carefully containing them have been stripped away. The carbohydrates are your polymeric sugars, so starches and various disaccharides that you get from milk and from vegetables, from fruits and vegetables. So then finally, proteins. Despite a common misconception, meat is not the only source of protein. Proteins are needed for in life of every cell. You can live off bacterial pellets, and you will get plenty of protein. As well as they're present in all the plant cells, so in all the fruits and vegetables. And then nucleic acids, something that your dietitian will never talk to you about, but we do consume DNA and RNA in large quantities. Our stomach breaks it down, and our gut absorbs the nucleotides. And the main sources for nucleic acid, aside from our own synthesis, are unprocessed foods, so things that still look like they were alive recently. So meat and fruits and vegetables. And this is a diagram that shows biomolecules and the fact that overwhelming majority of them are biopolymers. So you guys can see that the carbohydrates are made up of individual sugar molecules linked together either into disaccharides or polysaccharides like starch. Lipids are simpler. They're usually present in our diet and in our bodies when we store them in our thighs in the form of triglycerides. So it's 3 fat molecules linked together by glycerol. Proteins are chains of amino acids. If you break down a protein into smaller chain links, which are still a few amino acids together, that's called a peptide. Nucleic acids, so RNA and DNA, are all composed of nucleotides. There's 5 different nucleotides: adenine, guanine, thymidine, cytosine, and uracil, which is only found in RNA, while thymidine is only found in DNA. As well as things that biochemistry will deal that you will deal in biochemistry, such as high energy compounds like ATP, which are basically modified nucleotides. They also have the adenosine molecule that the adenine in DNA and RNA carries. They also have the ribose. It's the RNA type of ribose, not the deoxyribose of DNA. And in addition, they carry 3 phosphates in the end. And the third phosphate, the connection between the third and the second phosphate is very unstable. It breaks easily, and when it breaks, it releases energy, which is used to power overwhelming majority of biological reactions. So to summarize, biopolymers are DNA, RNA, proteins, as well as carbohydrates and lipids. DNA in our cells is used to primarily store information. The bacteria don't bother segregating into a special compartment. They just keep it around with everything else in this giant circular chromosome. The eukaryotes put it nice and tidy into its own special organelle called the nucleus, and the access to the nucleus is very much restricted. To get the information out of the nucleus and into the cytoplasm, where it can be translated and made into proteins, RNA is transcribed from DNA. So the main role that you see RNA in a cell is for information transfer as messenger RNA. However, RNA is critical. It's critical for catalysis as well. In fact, the ribosomes, which are the small molecular machines that translate RNA into proteins, are overwhelmingly RNA themselves. In fact, they function reasonably well when every single protein subunit from a ribosome has been stripped and only RNA is left. And then finally, proteins do everything in the cell. They catalyze all of the biological reaction. They provide structure. They allow for cells to have motion. They regulate various processes. They're secreted for signaling. They do it all. But it is the DNA that controls what they do and when. Now, carbohydrates and lipids sort of get the short stick in molecular biology because we're not going to talk about them. I'm going to just say that carbohydrates are primarily used for energy storage, which they're also used for structure in plants, etc. Lignin inside trees. And lipids, again, we will just say that they're primarily used for energy storage because they're excellent at it. But keep in mind that they are also important in signaling. However, our cells directly encode protein information inside of the DNA. We do not encode carbohydrate or lipid information. Instead, we make proteins to make the carbohydrates and lipids. So the connection of how these macromolecules are regulated is a little bit more removed. So, as I said, in this class, we're going to talk about DNA, RNA, and proteins, how they're made, and what things they can do. Before we go into DNA and RNA, however, we need to talk about chemical bonds and the principles that are key for allowing our cells to function. There's 2 types of commonly found chemical bonds in biology: the covalent bonds, which are strong, and the ionic bonds, which are weak. The covalent bonds is the way the actual units of all the biomolecules have been made to begin with, and this is how they're strung together. In a covalent bond, the electrons of the 2 atoms bonding are shared equally between the two atoms, creating a nonpolar cloud where the electron spends exactly the same amount of time in both areas. Though you have to understand that some atoms are more electronegative and others they're more electron hungry. So in some situation, there will be a slight shift in how long an electron spends next to one nucleus as opposed to next to the other. And we'll talk about it a little bit later. But you can see on top of the slide that DNA, molecules of DNA and RNA are linked together by covalent bonds, and those are the phosphodiester bond between the ribose and the phosphate. The reason they're called phosphodiester is because phosphate makes an ester oxygen bond on both sides. And as I said, covalent, you need to remember that covalent bonds are relatively short bonds and they're very strong. It will take a lot of energy to break a covalent bond for an enzyme. So they're structural things. And just like DNA and RNA are linked, subunits are linked together by covalent bonds called phosphodiester bonds, proteins are linked together from chains of amino acid via peptide bonds, which is the bond between the amino group of an amino acid and the carboxyl group of the next amino acid. Now, the ionic bonds, also known as salt bridges in biology, is a situation where one of the atoms involved is so electronegative that it flat out takes the electron. Surprisingly, that does not make for a very stable linkage because one of the atoms now acquires a positive charge, and the other acquires a negative charge because it has one extra electron. However, even though they're weak, they're extremely useful in biology. Now, some of the amino acids, in fact, 3 amino acids, there's 3 positively charged amino acids and 2 negatively charged amino acids, and about 7 amino acids who are weakly polar. And ionic bonds can be created between all of them. So in the picture that just appeared, you can see that the ionic bonds between the negatively charged glutamic acid residue and the positively charged arginine residue stabilize an inside of a protein, allowing for its shape to be formed. Another important famous ionic bond that occurs is when a salt that's just been dissolved in our cytoplasm, in this case magnesium, which carries a partial positive charge, carries a positive charge, creates ionic bonds with water and ATP. So the phosphate group, especially the third phosphate group of ATP, is negatively charged. It has a bunch of oxygen that have hoarded their electrons and are carrying a negative charge on them. Magnesium is positively charged. As a result, an ionic bond is formed between ATP and magnesium, and that does not destabilize the covalent bond between the third phosphate and the second phosphate. Now, if the water was allowed, which carries a partial, which is a dipole and carries a partial positive and negative charge depending on the location inside the molecule. If water was allowed access to that third phosphate and the magnesium wasn't there, that would allow the oxygen to attack, the hydrogen and oxygens to attack the third phosphate covalent bond linkage and break off the third phosphate, releasing energy unnecessarily and converting ATP into ADP plus inorganic phosphate, a process called hydrolysis of ATP. If you would just take ATP and put it in water, it would hydrolyze completely within hours. However, our cells have magnesium inside of them, and that prevents ATP hydrolysis inside of the cells and make ATP stable enough to be able to be synthesized, then transported to location where it's going to be used, and only then broken down. In fact, ATP proteins that use ATP to catalyze the reactions they would like to catalyze specifically have a water-less binding pocket for ATP. When ATP inserts the pocket, the water is squeezed out, allowing the magnesium is removed, allowing for magnesium is removed, allowing for the water to attack the molecule and for ATP to be hydrolyzed. So that's one of the reasons I just presented you a nice reason why biology doesn't just use covalent bonds in its chemistry. It also uses ionic bonds, even though they're weak. The other bonds that it uses is hydrogen bonds, which is electrostatic interactions. We talk about in the next few slides. And even though covalent bonds are strong, we need the weak type of bonds because the weak types of bonds, such as ionic and hydrogen, as well as hydrophobic interactions, allow for transient interactions between protein or nucleic acids. They allow for folding of an amino acid chain into a complicated three-dimensional shape. And breaking and reforming these weak bonds uses far less energy. You don't have to have a stable bond in order to do complicated chemistry. Now, let's talk about the weak chemical electrostatic interaction. The most important electrostatic interaction occurring in living organisms is the hydrogen bond. Now, the hydrogen bond is possible due to the fact that water is a dipole molecule. Unlike its buddy methane, which is 4 hydrogens bonded to a carbon, the water molecule carries a partial negative charge on the oxygen and a partial positive charge on the 2 hydrogens. Because the charges cancel each other out, because there's both positive and negative charges, overall, water does not carry in charge. You cannot put a magnet to water and it's going to go to it, right? But depending on which side it orients itself, it can make hydrogen bonds with each other or other compounds. So the asymmetry neutron distribution makes this molecular polar because the electrons spend more time around the oxygen which likes them, as opposed to the weaker, friendlier hydrogens. So this is demonstrated by this cartoon. So the electrons inside water are symmetrically distributed, making water molecule a dipole, because oxygen is very electronegative. So in this blue picture, you can see that both hydrogen and oxygen are forming a covalent bond, and because sharing is caring, and because that's what a covalent bond is, they share electrons. They both exchange the electrons. And the electrons in a normal covalent bond are supposed to spend the same amount of time on both sides. However, oxygen can't quite let go, and it attracts electrons more strongly than hydrogens, and so the electrons spend more time around the oxygen creating this dipole situation. So the positive charge on the hydrogens and a partial negative charge on the oxygen. Because the interaction is based on charge, hydrogen bonds are classified as electrostatic interactions. Hydrogen bonds by themselves are relatively weak. They're about 1 kilocalorie per mole, so even weaker than ionic bonds. However, there's a lot of water in the cell, and many interactions can be combined to a very large effect. In fact, geckos use these electrostatic interactions, which are weak by themselves, but if you have many interactions on one paw, allows gecko to move on vertical surfaces and even upside down, simply by electrostatically interacting with the surface it's crawling on. And because water will form hydrogen bonds with itself, because every oxygen inside the water carries a partial negative charge, and every hydrogen a partial positive charge, it makes water sticky to each other. So that is why water has such an amazingly big range of when it's liquid. It freezes around 0 Celsius and it boils at 100. Overwhelmingly, all other compounds have a very, very short fluid state because they don't have these dipole interactions to allow for the liquid state to occur. And why do we bother talking about water? Even though we just talked about complex biomolecules, is that water is crucial for life. It makes up 70% of your cell's volume. It also makes 70% of our Earth's surface, and one of the things that scientists look when they look for life on a different planet is presence of water. And because it makes overwhelmingly All processes inside a cell occur in an aqueous environment, and so water influences everything. And hydrogen bonding is also very important for both stability and interaction of different proteins and nucleotides. So water doesn't just form lattices with itself, creating its liquid state. Hydrogen bonds will affect other molecules in solution. So if you have salt, which is an ionic bond compound with chlorine carrying a negative charge and sodium carrying a positive charge, water will interact with the salt atoms and break them down, allowing for salt to dissolve in water. All other polar molecules that are partially charged, sugar, a lot of the proteins that have partially charged surfaces because of the partially charged amino acids that will interact with water. That is why these things are soluble in water. And water is a very good solvent, which means that it can hold as many other many other polar molecules at high concentrations. So our cytoplasm contains so many molecules, it's basically viscous. And that's possible because water is a dipole and carries a partial charge and will accommodate and include other polar partially charged molecules. So with this, I wanted to show you guys an online video, a TED Talk, which is 5 minutes. Let me get it here. Which talks about bio. So what I wanted you to take away from the video is that salt, table salt, or any other polar compound that carries partial charges on its atoms will easily dissolve in water. And it's nothing that the fat does that causes the fat to be separated from water. The oil will always segregate. It's just that the water's ability to bond with itself pushes out and its inability to bond with fat, because fat does not carry a charge. It's just carbons and hydrogens, and there's no partial charge on them at all. They're very perfectly beautiful covalent bonds that share and care and share everything nicely, and thus don't have a dipole moment. As a result, oil, when mixed into water or into a cytoplasm, will form droplets and stick to each other. That's how membranes are formed, but also amino acids that have a lot of the carbons and hydrogens in them, a lot of saturated carbon-hydrogen bonds, will be unable to interact with the water, and thus will be forced to stick together, causing the hydrophobic effect. Now, the hydrophobic effect is super important for folding of proteins because it allows for a two-dimensional protein to acquire three-dimensional structure that has an oily hydrophobic inside. And more than a third of amino acids used in proteins are hydrophobic and thus will not interact with water, instead will pull it away. Also, some protein binding to each other can be hydrophobic as well. So let's summarize here. The chemical bonds that are important in biology are the covalent bonds and the ionic bond. Those are real bonds. Now, a hydrogen bond, even though it has the word bond in it, is more of an electrostatic interaction. And electrostatic interaction via hydrogen bonds is super important because polar groups and biomolecules can interact with each other or with water and form these transient connections that allows proteins to catalyze reactions, or DNA to form, or RNA to form complex three-dimensional shapes. It allows it to act as a catalyst as well. Also, as this last sentence says, the hydrogen bonds between biomolecules compete with the hydrogen bonds that the water that surrounds everything in a cytoplasm forms. So an interaction between the biomolecules needs to be more stable than the water, or the water needs to be excluded by some way so it doesn't interfere in that particular hydrogen bonding process. Furthermore, dissolved ions such as magnesium are important for stabilizing for stabilizing negative and positive partial charges inside amino acids or inside nucleic acids. For example, here the green picture shows an RNA molecule being folded, and the green little balls that are found in different locations of the RNA molecule stabilize its three-dimensional shape. So ionic bonds are formed when charged chemical groups, those that act as either an acid or as bases, form electrostatic interactions. And this can be combined, both hydrogen bonding and ionic bonding and hydrophobic effect and van der Waals interactions, which are physically a bit more complicated than I care to discuss here, allow for proteins to both interact selectively, which means they can choose their own particular partners and reject others, and dynamically, which means that some partners, once they bind, are then also able to release, which is really important because proteins, especially one class called the enzymes, are catalysts. I mean, overwhelming protein, that's what they do. They're not used up in biochemical reactions. They're just there to make the process go more smoothly. So they need to be able to bind each other and then release and continue going on to the next target. So this image shows you the hydrogen bonds between the carboxyl groups and the OH groups inside a protein, the ionic bonds between, say, the arginine or lysine and glutamic acid side chains of an amino acid, of a peptide chain, as well as hydrophobic interactions between hydrophobic side chains of an amino acid, such as valine, leucine, isoleucine, phenylalanine, and all those big guys that have a lot of carbons and not that much oxygen. And these interactions allow proteins to fit like a lock and key model, because these are still very, very short range interactions, and the proteins actively hug each other when these interactions occur. One of the important examples that you have faced in everyday life is the ability of the antibodies that are produced by the immune system of your body to interact with antigen. Antigen is a umbrella term saying it's a thing that sticks out of a foreign invader thing. So in this case, the picture shows a virus. Let's say it's a friendly rhinovirus and not HIV. And you can see the red guys have bound to the virus, and that their binding to the antigen, and then their binding to each other will allow the virus to be clumped up and mopped up by the white blood cells and removed from our body, allowing us to recover from our cold. And as interactions go, actually, antibody interactions between an antibody and antigen is one of the more stable ones, because they stay on their target, on the antigen, once they're recognized for a long time. So that property of an antibody to recognize something else, an antigen, is not just used by our immune system to defend us against viruses, but also used in different protein technologies in order to recognize different proteins. You raise an antibody against. So, as I said, the proteins are able to interact both selectively and dynamically with other proteins because they use electrostatic interactions and not covalent bonding to form. And because the more electrostatic interactions two proteins can come up with, the better and more stable their bonding will be. So you can see here protein A has a few side chains poking out of it that are capable of an electrostatic bond. So it can form 3 bonds with A and B, but the Brownian motion is that does not is not enough stability in an aqueous environment of a cytoplasm to make this bond very permanent. So A and B will release and float away. Same goes with A and C. Somewhat more bonds are formed, but still not enough to outcompete the interactions with water that both A protein and C protein are experiencing. However, A and D form lots of electrostatic bonds with each other, most likely hydrogen bonds maybe hydrophobic bonds, that allows them to stick together and become binding partners. And an example why this, because of competition with water, it's really important for these interactions to be reversible, because this allows interactions to be dynamic, because we need dynamic interactions in order for motor proteins to move along the cytoskeleton of the cell, in order for channels to function, in order for proteins bind and release, in order for receptors to interact with their ligands. Many, many, many things depend on this both selective and dynamic interaction between proteins, which is all due to electrostatic interactions, the chemical bonding that they are capable of. And so this concludes the biomolecule and bonding part of the first lecture, and I will start recording the enzymatic part of the lecture now. Stay tuned.